Hello, good afternoon everyone. My name is Richard Pine. I'm Director of Corporate Communications at SUMMT Therapeutics, and today I'd like to talk to you about what we've got going on with the company and highlight some of the milestones that we are looking forward to over the coming 12 months. Just our legal disclaimer, we are listed both here in AIM and also on NASDAQ, and must refer you to the disclaimer and also our filings with the Securities and Exchange Commission in the US. So as a company, we're a biopharmaceutical company. We're focused on two disease areas. The first is Duchenne Muscular Dystrophy, where we've established a leadership position based upon some pioneering technology that we brought out of Oxford University. And I'll talk through the progress we've made to date and highlight what opportunities we've got coming up over the coming 12 months. That will focus predominantly around our lead candidate, which is progressing into a phase 2 clinical trial. We're also developing a novel antibiotic for the treatment of C. difficile infection. Again, this program has made fantastic progress over the last 12 months and culminated in the reporting of some phase 2 proof of concept data late in 2015, and leaves us in a very nice position as we now progress this asset into phase 3 trials and decide how to maximize commercial value from this. As I mentioned, we are dual listed both on AIM and on NASDAQ, having completed our US IPO in March 2015. So our pipeline breaks down into two core components. We have this concept we call utrophin modulation, and this is the pioneering technology I mentioned. This was originated by Kay Davis at the University of Oxford, who had spent her life working on this as a concept for a disease-modifying approach for DMD, and I'll talk more about that in a moment. But as you can see, we've got three broad strands. We've got our lead candidate, which we call SMMT C1100, but we're also building a strong pipeline behind that. So it means that we have established ourselves as the leaders in utrophin modulation, and if this concept works, means we are very well placed to take advantage of that. And also we hope improves the lives of the patients and their families suffering from this condition. Our C. diff antibiotic is called ridinilazole, and again that's now progressed through phase 2, and I'll report on some of the data, which is why we're so excited about its potential as again a new treatment option for this condition. Firstly, just moving on to DMD, and just to give you a little bit of background as to what is this condition. Now, this is a fatal muscle wasting disorder. It's caused by different genetic faults in the gene that produces a protein called dystrophin. Now, we all have dystrophin in our bodies. It's what allows our muscles to function healthily. Boys and men with DMD are unable to make dystrophin. What that means is that over time their muscles break down. It means they are unable to walk by their teenage years. Means they will need full-time respiratory support by their late teens into their early 20s, and ultimately once they've been left paralysed it will prove to be fatal by their late 20s. There is currently no cure for DMD. It's classified as one of these orphan diseases, which means there's a relatively small patient population compared with more common diseases such as diabetes. There's estimated to be around 50,000 boys and men with DMD in the developed world. And that classifies it as an orphan disease, and that means we get a number of benefits. You can get additional regulatory support. You can also get extended patent terms. In addition to that, you can also command premium pricing. And indeed, a number of orphan drug products that have made it to market have been classified as blockbusters, meaning they sell in excess of $1 billion per year. And we believe that this is a very exciting opportunity because it's one of the larger orphan diseases, and we have an approach that we think will be applicable to all patients. I just wanted to briefly mention what we're trying to do and go back to sort of the mechanism behind the disease itself. So I mentioned that the boys and men with DMD are unable to make this protein called dystrophin. However, the body produces another protein called utrophin, and what we're showing here in that top line, these dots, is the length of a muscle fibre, and that's where all the protein is being expressed. In a normal fibre, you have utrophin there first of all, but then as a fibre matures, the utrophin is turned off by the body, and then dystrophin comes along to perform the same key structural role, and that maintains healthy muscle function. However, in boys and men with DMD, they can make utrophin like we do. However, they can't make any dystrophin, and that leaves these sites unoccupied. That leads to their continual progressive muscle wasting, and ultimately will prove fatal. What we're looking to achieve is a small molecule approach whereby we will look to interfere with that mechanism and keep utrophin switched on all the time, so it can compensate for the absence of dystrophin in a mature muscle fiber. And we're doing that using conventional small molecules, a very traditional pharmaceutical approach. I'm going to just briefly talk about our lead candidate called SMT-C1100. And this is an orally administered drug. Indeed, it's actually formulated currently like a cough syrup. So the boys would take around 20 mils of product, and very easy to administer, ideal given that we are dealing with a pediatric population as well as men who have difficulty swallowing as the disease progresses. We've shown in preclinical that this drug works. We've been able to increase the amount of protein. We've been able to show a clear functional benefit in disease models, and we're now looking to show that effect in patients. We've completed phase 1 trials where again the drug has been well tolerated both in healthy volunteers and also in patients with DMD. As I mentioned, it's one of these orphan drugs, and we're in a very good position there because we have orphan drug status granted both in Europe and in the US, while our IP position is very strong, having now got granted composition of matter patents both here in Europe, in Japan, and also in the United States through until at least 2027, with the potential for patent term extensions there as well. So that leaves us in a very good position from a commercial stance. The next step, so for this program, is to actually show it works in patients, and we've now received the approval from the UK regulators to start a phase 2 proof of concept trial. This will be the first opportunity where we will look to see if this drug actually works, and that's going to be a significant milestone for this program. If we can demonstrate that we are able to modulate the utrophin expression and have a functional benefit, then that's going to add a lot of value. We believe will be a clear value inflection point for this product. We are due to start that trial imminently, and we look forward to reporting the first set of data from this before the end of this year. So again, it's not too far away before we reach this significant milestone. Now, we're very excited about C1100, but in the time that we've been focusing on that, we've also been developing our pipeline because we can already see ways of improving upon that. So that will give us the opportunity to have the first drug to market, give us the best drug candidate, and then also bring these other ones through behind it. And what we're working on here are twofold. Firstly, we have our second generation candidates. These are similar to C1100 in terms of their structure, but just have improved properties. And again, we've demonstrated that in preclinical studies, and we're working now to select one of those to take on into clinical trials. Behind that, though, we've maintained our relationship with the University of Oxford, where this whole concept originated. And we formed a strategic alliance with them back in 2013, which we extended at the end of 2015. And that's working on future generation compounds, some of which work in a similar manner to SMT-C1100, but also with new mechanisms. So again, it's really strengthening our position in this field. If this concept works, Summit will be in a very strong position to benefit from that. There is currently no other product in clinical trials that has the potential to modify this disease in all the patients. I'm now going to sort of change gears a little bit and talk about our second program, and this is quite different. This is an antibiotic for an infection caused by the bacteria Clostridium difficile. Now, you may well have read quite a lot about C. difficile infections in the papers. There's been outbreaks of it within the hospitals here in the UK and elsewhere across the globe, and it is a major healthcare issue. It's estimated that in Europe and North America there's well over 1 million cases per year. It isn't just a sort of a mild disease where you get a bit of sort of upset tummy or anything. This is very serious, and in a number of cases will prove to be fatal. As a result, it's got a high economic burden. It's estimated that in the US alone, acute care costs are at about 4.8 billion per annum. So that's a substantial problem. Now, the issue here is something called disease recurrence. So what that means is patients get an outbreak of C. difficile, they're then treated, but the treatments that they currently take leave them very susceptible to have second and third episodes of the disease. And indeed, a patient who has their first episode of C. difficile infection has a 25% risk of having a second episode when treated with current agents. If they have a second one, that risk goes up to 50%, and you end up in this vicious cycle where patients are becoming more and more susceptible to the illness. The reason for that is they're treated with what we call broad-spectrum antibiotics, very conventional. They're very good at treating C. diff, but they actually cause a lot of collateral damage to your gut microbiome, and that provides a protective effect against the illness. So ideally what you're wanting to do here is not treat it with more broad spectrum antibiotic, but develop a selective antibiotic that only targets the C. diff and allows your gut microbiome to recover and provide its natural protective effect. Indeed, that's what SUMMit has been working on. We've developed an antibiotic called ridinilazole. This again is a small molecule, very conventional in its approach, but it's highly potent for C. difficile, but most importantly, highly selective. It causes very little damage to the wider gut flora, and that's a result of which we believe this could have a profound impact both as an initial treatment, but also reducing rates of disease recurrence. We completed the phase 2 proof of concept trial at the end of 2015, and I'll show you that data in a few moments. And we expect to report more data from that during the first half of this year. In addition to which, we benefit from some other things. In a similar way to the DMD being classed as an orphan drug, governments are now recognizing the problems of not having new antibiotics coming through. In the US, they produced the GAIN Act, generating antibiotic incentives now, and that came into effect in 2012. And that provides us again with additional regulatory support and market protection, having been granted QIDP status by the US Food and Drug Administration. We've also been designated fast track status, which should allow us for a faster regulatory pathway. I must acknowledge, though, that the progress we've made has been supported by the Wellcome Trust through 2 grants over a number of years, which has allowed us to get to the end of this phase 2 trial. So this trial in a bit of detail. This was a fairly conventional placebo-controlled sort of a blinded study against the current standard of care called vancomycin. In this study, we recruited 100 patients, half of which received our drug ridinilazole and half of which received vancomycin. And we were looking at an endpoint known as sustained clinical response. And quite simply, that's just looking at the rates of initial cure at the end of treatment, which was 10 days, and then also how many patients had a recurrence of the disease within the 30-day monitoring period afterwards, because that's typically when a patient will have an outbreak of repeat C. difficile infection. And what we saw from this study was quite remarkable. Vancomycin, which is the current standard of care, had a sustained clinical response rate of 42.4%, while ridinilazole was over 66%, a 24% difference, and that was showing clear statistical superiority. And what was significant about that, not only was there this clear separation, was what was actually driving it, and that was a numerical difference in recurrence rates. So this was showing we were addressing the primary clinical issue with this disease in this phase 2 trial. So this was hugely exciting for us, and we're now very much looking forward to seeing this product move on into phase 3 trials. I think what's also important, I just wanted to highlight why we believe we were having this profound effect in the phase 2 studies, because we produced a raft of preclinical data which showed the potential of ridinilazole over a number of years. And this one piece of data I wanted to highlight was its effects on the gut microbiome and the bacterial families that comprise that. First of all, I just wanted to highlight its potency against C. difficile, and I'm comparing here ridinilazole with 3 agents that are on the market: metronidazole and vancomycin, which represents around 95% of the C. diff market, with fidaxomicin, a new agent, as a sort of a niche within there. And you can see they're all fairly potent with each other. But this really highlights the issue. These other agents, where there's lots of red being shown here, means they're very potent. And what they're doing is damaging that gut flora. These are all broad families of bacteria, and you can see they're all having a broad antibiotic effect. When compared with ridinilazole, you can see we're having a minimal effect against this. So that showed that we're combining potency with selectivity, and that was what was leading, in our view, to that phase 2 result, which I showed you a moment ago. We're hugely excited about the potential of this. We're now looking at ways of taking this forward and maximizing the value and preparing to enter phase 3 trials while we undertake that exercise. Our financials are relatively straightforward. We're a loss-making biotechnology company, so the key figure for investors is usually our cash. And here we can see we reported a £22.2 million balance in our Q3s, which is the end of October. We are listed on both AIM and on NASDAQ. So we've shown here the AIM price and market capitalization of 76 million based on that closing price. I think in our view, we've got a number of opportunities coming up over the next 12 months and beyond, which can act as significant catalysts to increase the share value for the company. So what are those catalysts? Well, firstly, it's going to be that first readout from the phase 2 proof of concept study for our lead utrophin modulator drug. We'll be looking to report on that during the second half of this year. In addition to which, we'll be reporting more data from that phase 2 proof of concept trial for our C. difficile antibiotic over the first half of this year. So there's going to be good, steady news flow from both our programs, and leaves us in a very strong and exciting position as we watch those progress to the next stages of development. I'm here all day, so please do come and visit us at stand 26 and ask all your questions. I'm mindful that I'm keeping you all from your lunch now, so I think with that I will conclude, and thank you very much for your attention.